to your first home video about cell division. Three key things to know about cell division. One, all cells contain chromosomes, which we defined in class today or yesterday, that contain DNA or genetic information that codes for all traits in the cells. Cells that respond and react to their environment using cell signaling. We've learned that before. Think reception, transduction, and response. And we know that organisms have specialized tissues or organs or cells or proteins to do very simple tasks like regulate our temperature, our respiration, our metabolism, etc., etc. We're not going to look at these two videos today. Maybe that's something we will do on Friday. Whoops. So, why we care about cell division? Because once upon a time, you each started off as a single cell, and you ended up as, 9 months later, a couple trillion cells with very specialized functions and tissues and cells. The development of an organism always begins with a fertilized egg and a sperm cell, which means that this cell has two sets of chromosomes or DNA. We call these organisms diploid organisms. Now, over the next 9 months, a fetus is going to grow in terms of both the number of cells and size, but also how those cells are located and the types of cells that we see. The cells in your muscle cells are different than the cells in your brain or your eyes or your tongue. For the most part, divisions make exact perfect copies. So one skin cell becomes two skin cells. In development, cells as they divide begin to become differentiated or determined. So these cells become eye cells, these cells become ear cells, these cells become nose cells. Differentiation occurs much later in fetal development, and even into puberty and adolescence, differentiation is occurring as you become mature adults. So cell division isn't just making more cells, it's also sometimes increasing the types or numbers of cells. Now, in terms of mature cell bodies, over here we have a bunch of different cells and then their turnover time. How quickly do they go through mitosis? On average, the human body replaces itself every 7 years, but like your body temperature being 98.6 degrees Fahrenheit, it's a simplification. It's not that straightforward. Parts of your body are never replaced after the tissue or the organ is developed in a fetus or young child. This means you lose it. If you break it, you lose it. You're never going to get it back. These are irreplaceable and indeed the hardest body parts to fix. So, for example, lens cells. If you become blind, that is very problematic. Central nervous system. That's why if you damage your spine and become quadriplegic or paraplegic, that is often permanent. These are parts of your body that are just permanently intact and cannot be broken because they don't have a replacement rate. They don't have an ability to fix themselves. Other parts of your body are constantly replaced, and they have so much usability and weathering and impact from the environment that they have to be constantly replaced. That can be based on your environment, your genetics, your age, your health, your diet, etc., but they are replaced often. A lot of your intestinal lining is replaced often. White blood cells for immune system, your lungs, again, lots of environments, taste buds that have a lot of skin epidermis interaction with environments or damaging things. And then parts of your body stop growing or are replaced much less often as you age. This might be due to sex characteristics, so testes and ovaries or other reproductive characteristics, or sex hormone levels, your testosterone, your estrogen. These tend to peak in your mid-20s and early 30s, and then they drop off. So, for example, spermatogenesis, where are you? Male gamete sperm lasts for 2 months. Generally, younger adolescent males and young adults make more sperm than older males based on, again, testosterone levels. So, why do we divide? There are three main reasons why human cells divide. One is replacement. Cells need to replace dead or aged cells in order for the tissue and organs to remain optimal. So basically, this is a proactive step in which we are replacing cells that might be too old with something that is new before it breaks. The rationale is that over time, our cells are used and abused. They experience mutations, cellular damage, reduced efficiencies. You go to Florida for a week and tan all day long. Your epidermal cells need a break. They need to be replaced. So again, examples of this would be your epidermal skin cell, your epithelial intestinal cells, your liver cells that are breaking down toxics and poisons, your taste buds, which are interacting with all of your foods. These cells need to be replaced before they break so you can taste, detoxify, absorb nutrients, protect your body from the sun. Now, other times cells divide because we're trying to experience growth. We're actually seeing an increased number of density or number or frequency of the cells in our body. And this is because, based on the environment and signals, we might need tissues to be bigger, stronger, more different. So myocytes, after you lift, hypertrophy, puberty and secondary sex characteristics, hair follicles, especially if you are developing or you're in a colder environment, red blood cells in response to exercise and elevation, so people can have more red blood cells in their body if they exercise more or go up to the mountains. And then finally, repair. At times, we need cells to divide and help repair broader systems such as tissues, organs, or organ systems because things are broken or are breaking. We didn't do a good enough job with the replace. Now we have to repair. While individual cells might be healthy, the reason why division occurs is because tissues or organs need to be reinvigorated or repaired because they are broken. Good examples of this are osteocytes and osteoblasts when a bone break, liver hepatocytes after toxin are ingested, such as alcohol or narcotics, epidermal cells after a cut. So good examples would be your epidermal cells are repairing your skin tissue after a cut, or your bones are healing after a break. So 3 big reasons why mitosis is occurring every day in your body. Now, there are 4 main actors that we care about when it comes to cell division. We have chromatin, and that is going to be DNA with proteins that create chromosomes. Their importance for mitosis is that they need to be duplicated perfectly for the 2 cells to get the same exact information. 1 skin cell is going to become 2 skin cells. We need both skin cells to do the same thing. During mitosis, what we call euchromatin is condensed to heterochromatin. So during mitosis, we will, let's see if, I'm going to go through all of this so we can just see this picture right now. Where'd it go? Shoot. Sorry for the delay. Where did that picture go? Oh, it's here. That's why. Shout out to Liam. And who else is there? Lucy. I can recognize Lucy because they're a cute golden retriever. They're the best. All right. There we go. Okay. So now we can see heterochromatin being condensed from euchromatin. And this is meant to represent the duplicated copies. 1 copy, 2 copies. More on this tomorrow in class in terms of what I mean by that. Now, in terms of centrioles and centrosomes, these are the organizers. These are the movers. They are the orchestra, the conductor, the engineer of cell division. Centrioles, which are made up of 2, make centrosomes. They produce microtubules to stimulate division. More on that in a second. And the changes that they experience as they move to the poles to help spur division. So these are our centrioles. There's 2 of them that make the centrosome. They move to the poles, and then they use proteins to help split apart our DNA into 2 exact copies. And speaking of microtubules, these are small repeating protein chains that easily grow and are easily broken down. They sort of look like straws or rope. They attach to duplicated chromatids and help split the pair, and there are changes as they grow outwards, and then they retract this towards the centrioles. So they grow outwards, attach, almost like catching a fish, and then reel it back in. And then the last thing we have to talk about are kinetochores. These are protein complexes that organize and attach the 2 duplicated chromatins, or chromatids. They enable synchronized and controlled separation, and they break apart during metaphase. So these are our kinetochores. They occur at a region called the centromere, and they attach the two duplicated copies of each chromosome to each other, and then attach on both sides to the kinetochores, allowing us to separate. So again, a lot of vocabulary words with mitosis. We have chromatids or chromosomes, which refer to DNA and proteins. We have centromeres and kinetochores, which are at the center. They hold things together. We have the centrioles, which make microtubules that attach to the centromeres or the kinetochores. There's a lot of vocabulary words, so feeling confident with identifying, labeling them is going to be really important. That is it for our first video. I hope this has been helpful. Come into class with questions.